Glucose is oxidized in your body to produce energy. Now you could oxidize glucose on the benchtop. You could burn it in oxygen, the combustion of glucose. You'd burn glucose in oxygen, you'd form carbon dioxide and water. Energy would be released. In your body, the same thing happens. You oxidize glucose, breathe in oxygen to react with the glucose, you breathe out carbon dioxide and water. So the products and reactants are the same. You start with the reactants, glucose and oxygen. You form the products, carbon dioxide and water, whether you do it on the benchtop or in your body. So thermodynamically, the energetics are the same, basically. Free energy, entropy, enthalpy, they're state functions. They only depend on the initial and final state. I have glucose and oxygen, and I form carbon dioxide and water. About the same states, the same energy is released. Whether I do the reaction burning it in oxygen on the benchtop or at body temperature, catalyzed by enzymes. The same amount of energy is available for your body as you would have from the benchtop oxidation. In your body, the oxidation occurs more like a battery. The oxidation occurs in one compartment and the reduction in another compartment, like an electrochemical cell. The compartment where the oxidation occurs is inside the mitochondria of your cell. A mitochondria is a small organ within your cell. The mitochondria is surrounded by a membrane made up of phospholipids. Phospholipids are molecules that I've drawn here with a blue hydrophilic end, that means an end that is attracted to water, and I've drawn a yellow hydrophobic end that fears water. Now, in solution, the hydrophobic ends associate, and the hydrophilic ends are out towards the water. So these phospholipids form a membrane spontaneously, simply by associating their hydrophobic ends together and exposing their hydrophilic ends to the water. Inside the mitochondria, the oxidation of glucose occurs in the absence of oxygen. It's a formal oxidation. I remove a pair of electrons. So the glucose transformed into carbon dioxide, electrons are removed, oxidation, and protons. Those electrons and protons are transferred to the membrane by small molecules that transport them there. And I've drawn the small molecules here as this oval. They're transported to membrane-spanning proteins. These are large molecules, I've drawn them as ovals here, that span the membrane. They go from one side of the membrane to the other. So small molecules take our products of our oxidation, our electrons and our protons, to the membrane-spanning protein, and the protons are passed through the membrane to the outside, the cytoplasm of your cell, outside the mitochondria. The electrons are transported to another membrane-spanning protein, and on this membrane-spanning protein, oxygen is reduced and combined with protons to form water. So, as an electrochemical cell, you might call this the cathode of the electrochemical cell. This membrane-spanning protein acts as a cathode where the reduction occurs. Now, you can see that protons are consumed, H pluses are consumed when I do the reduction here, and they're actively pumped outside the mitochondria during this oxidation step. So what you have is a pH gradient across the mitochondria. The pH gradient is used to power the production of ATP, adenosine triphosphate, the energy storage molecule of your cells. It's used to power them as protons are allowed back in with the gradient going downhill from high concentration of protons to low concentration of protons. That is coupled with the phosphorylation of ADP, adenosine diphosphate, to adenosine triphosphate. So oxidation of glucose occurs by a very elegant method, like a little electrochemical cell, within your cells using the mitochondria as tiny batteries.